Hey guys, today we'll be learning about the staining techniques. So your second lab exercise has gram staining technique. Gram staining is a technique used to differentiate the gram positive bacteria from gram negative bacteria. As I always said, there are multiple staining techniques in microbiology diagnostic labs. The very first technique that anybody can use is a simple staining technique. Simple staining is you just use one basic stain to quickly take a look at the shape and size of the cells. But that will not give you much of an information. So that is the reason we need to go for differential staining technique. As I already explained in your chapter 3, I believe, differential staining technique uses two different kinds of stains. Okay? So you have one stain that is known as a primary stain that is used and added to all the cells. You wash it off, you then go with another stain, you use the decolorizer to wash the stain off, and then you go with the second stain to differentiate the gram positive cells from the gram negative cells. Okay, so I certainly suggest, I highly recommend, and you must watch all the videos that have been uploaded on Canvas for the second lab exercise. Apart from that, you also should pay close attention to the procedures. I will be showing the demonstration now of the gram staining technique, but it's really important for you guys to understand why each and every reagent is added in the procedure. Gram staining technique has two parts. The first part is to make the bacterial smear. So what exactly is making a bacterial smear? You take the solid culture. So this is the solid culture where we have Klebsiella pneumonia growing on TSA plates, or you can also take the liquid culture. So this is Serratia marcescens, I believe, growing in the tryptic soy broth. So you can either use solid culture or liquid culture and add those cells on these microscopic slides. Okay, so this procedure is known as making a bacterial smear and making this ready for your gram staining procedure. Okay, let's just light the Bunsen burner. We have the flame going and we are ready to make the bacterial smear. So today I'm going to show you how to make the bacterial smear using the solid culture as well as the liquid culture. I have gram positive bacteria, Staphylococcus aureus, on the plate here, and gram negative bacteria, Serratia marcescens, as a liquid form. Okay. The goal is to add enough cells on the slide, on the microscopic slide, so that we can stain it and look those cells under the microscope. Okay, but the most important thing, very careful when you're picking the cells. You don't want too many cells on your slide, but at the same time you don't want too little. Okay, so as always, you have the inoculating loop, flame sterilize it. Wait for it to cool down. And pick few cells, very few cells. You don't want to have too many cells, because having too many cells would make your microscopic slide too crowded and you won't get good gram staining pictures. Remember, the goal is to be able to identify or differentiate between the gram positive versus gram negative bacterial cells on the slide. Think about a patient coming to a physician's office with an infection. The most important thing is the physician has to find out whether the infection is from gram negative bacteria or from gram-positive bacteria. Only then the right antibiotic can be given to the patient. So the very first technique would be, once you streak the plate, once the microbiologist would streak the plate, the next technique would be the gram staining technique. Okay? Alright, so you have the bacteria here, and because we have the bacteria as a solid culture, what we need to do is add a loop full of water so that you can dissolve the cells nicely in the water. Okay? The water need not be sterile because anyway we'll be fixing the cells and we'll be killing the cells, so we are not really growing the cells, so we don't need to really worry about water being sterile. You can use the DI water right on your tables. Okay? So again, water, you don't want too much. I am making sure I have only a thin film here. Okay? So I'm going to tap, 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 and make a circle. And as you are spreading the water, you can see that it is kind of cloudy, which is good because that would really make sure that you have your cells in the liquid, in the water. You want cells, right? If it is not cloudy, you will be a little confused whether you have enough cells or not. I can see that the liquid is cloudy, so I'm pretty sure the cells are right there. And now I'm going to sterilize the loop. Put it here and labeling the slide. So this is important, but at the same time, while throughout your gram staining technique, it's really important for you to kind of focus and ballpark on what side you are adding the cells. Because I might have, a label here, so I usually, what I do is I have like a marking on the other side, not on the side where I have my cells, but on the other side. So I labeled both my slides. One of the slides has the bacteria on it. I added the solid culture, wherein first I put a little bit of water and then nicely mixed all the cells so that the cells are not clumped up. And now we need to wait till all the liquid gets dried up. So the next step would be to heat fix the cells by running the slide through the flame gently. But before you do that, it's very, very important to let the slide completely dry. Now in the meantime, while I am waiting for my solid culture to dry out, what I'm going to do is I'm going to add the liquid culture onto the other slide. I have it here. So liquid culture, what's the difference between making a smear using a solid culture and a liquid culture? Because this is already in liquid form, you don't need to really add water. You can directly take the liquid culture and spread them on the slide. So always whenever you're dealing with liquid cultures, make sure you're really vigorously mixing them because the cells usually settle at the bottom. And then you open, you clean the lid, and holding it near the flame so I'm not contaminating anything. Right? By the time it comes to the room temperature, letting my loop cool off a little bit. Go and grab the cells. So tap it. And I am going to add a few more loops just to make sure I have enough cells in the liquid. Alright, so I think I have enough cells. Now I'm going to close my liquid culture, put it here, and spread the liquid culture on the slide. So this is the difference between when you're doing gram staining technique using a solid culture versus liquid culture. You don't need to add water when you're handling the liquid. because it's already in the broth. Alright, so let me just sterilize it. Put it here. Now you can clearly see that the solid culture on the slide is all dried up. Okay? When everything is dried up, when there is no visible water, it's ready to be heat fixed. So what we are going to do is gently run the slide through the flame for 2 or 3 times. 1, 2, and 3. That's it. This is good. Let me explain you a few concepts here. So what is the purpose of heat fixing the cells? The heat fixing step makes sure that actually the cells are sticking well to the slide, microscopic slide. That's one of the reasons why you fix the cells using heat. There is also another reason. You do not want to contaminate your microscope or any of the surfaces around. So that's the reason you heat fix it, so that the heat would actually kill the bacteria. What will happen if you run the slide many times for a long period of time? The bacterial cells would die, and the cells also would get distorted. They would change the shape. So that is really not good for the gram staining technique. And that is the reason whenever you're running through the flame, you have to be gentle, you have to be quick, and you have to flame enough. Okay? So you'll have to pass through the flame or through the Bunsen burner for 3 times without exposing the cells to a very, very long time. Okay? Alright, so my solid culture is ready, and you call it bacterial smear. So we made the bacterial smear using the solid culture, and now my liquid culture is almost ready. I don't see any visible liquid. It's all dried up. So there you go. 1, 2, and 3. Okay. I do not need the Bunsen burner now, so I'm going to turn it off and make the space ready for the gram staining technique. For the gram staining technique, you need the gram staining reagents, which are right here. Distilled water, because you'll be washing the stains. And you also need a timer. Procedure, it's very important to make sure you have all the reagents in the specific order. The first step would be to add the primary stain, which is going to be crystal violet. So I have it right here, and I make sure that I have enough for my gram staining. The second reagent is Gram's iodine solution, and I do have it here. The third solution is decolorizing agent. I do have it here. And the fourth reagent is the secondary stain or the counter stain, which is safranin, which is the pink stain. Okay? Right. The procedure, every lab follows a specific procedure, and we here follow 1 minute of the primary stain, followed by washing quickly with the deionized water, and then we add Gram's iodine for a minute. We wash it with water. We add decolorizer for a very, very brief period of 8 seconds only. Okay? And then we stop the action of decolorizer by again adding the distilled water, and finally we add the secondary stain, which is safranin, and we add it only for 45 seconds. Okay? Wash it with water, and then it will be ready to be viewed under the microscope. Before I begin, I would like to say a few things about why we are adding certain reagents here. So what's the purpose of adding primary stain? Primary stain is a crystal violet. It's a basic stain, and it can stain any cell, any bacterial cell. So once you add your primary stain, all the cells, whether they are gram positives or gram negatives, all of them should be stained purple. Okay? And then when you go and add Gram's iodine is a mordant. So what does a mordant do? A mordant is added so that the primary stain sticks to the membranes of the cells really well. So it forms a complex with the cell membrane, and it keeps the dye inside the cell better. That's the reason we add mordant. The third reagent is decolorizer, so that the primary stain is washed off from gram-negative cells but not from the gram-positive cells, because gram-positives have thicker cell walls, they retain the stain. Only if you add the decolorizer for a very brief 8-second time. Okay? And then you go and add your secondary stain, which is the counter stain or safranin, and at that point only your gram-negatives are able to take the pink stain, because gram-positives, they already have the darker crystal violet purple stain. So at the end of your gram staining procedure, gram-positive cells should look purple and gram-negative cells should look pink. But always remember, there are exceptions. There are many bacterial cells that will never take up any of these stains. They are atypical cells, especially, I don't know if you remember, I already mentioned about some of these pathogenic strains like Mycobacterium tuberculosis. They never can be stained using your simple crystal violet and safranin. You have to use a special stain known as acid-fast stain if you're looking to stain those pathogenic cells like the Mycobacterium tuberculosis. So that's an exception. And yet another exception, some cells, they are known as gram variable. When you try to stain them using crystal violet and safranin, some of them will take up the purple stain, some of them will take up the pink stain. So those kind of cells are known as gram variable. So always remember, there are exceptions, but majority of the times gram positive cells would look purple and gram negative cells would look pink. All right, let's start the procedure. We have the timer, we have the reagents, we have the distilled water, and we have the smears all ready. So you add the primary stain, completely cover the slide using the primary stain and start the timer. Put the stain for 1 minute and then you wash it nicely by holding the slide like this, and you wash it front and back with the distilled water. And as I always said, you have to remember on what side you have the cells. If you forget the side you have the cells, that will be a problem. So I'm washing both my smears, front and back, with distilled water. And that is the very first step of the gram staining. Second step is to add the grams iodine solution. So I have the grams iodine here. Once again, cover the slide completely with the grams iodine and leave it on for, okay, the 1 minute is done, and I am washing off the grams iodine the same way that we wash the primary stain front and back. And remember where you have your cells. Remember not to flip. Right, washing the slide front and back. Right, now the most critical step. In gram staining technique, this is really an important step, using the decolorizer. So decolorizer should be only used for a very brief period of 8 seconds, just to wash off the primary stain from the gram-negative cells and not from the gram-positive cells. What will happen if you use the decolorizer for a long time? The stain from the gram-positives and gram-negatives would be all coming off, and you don't want that to happen. So that's the reason. Use your timers and be very specific to use the decolorizer for only 8 seconds. Okay, my 8 seconds are done, and I am stopping the action of decolorizer by adding the water, washing it with water. I'm adding the decolorizer to the liquid smear also. Just for 8 seconds, and stop the action by washing it with water. Final step is to just go and add your secondary stain, which is a safranin. Okay, and the same way, completely flood the slide using safranin and start your timer. And now wash front and back with distilled water. Same way, I have the liquid culture smear. Wash front and back. The final step before your slides go onto the microscope is to dry the slide completely using the Book of Bibulus paper. This slide seems to have a little bit of dye, so I'm going to wash front and back again. So there you go. Have the slide, solid culture slide, and before you look the cells under the microscope, the most important thing is to make sure your slide is completely dry. And we do this by gently pressing the slide using the Book of Bibulus paper. I've noticed over the years that students do all this procedure, and when they are at this step, they would press this too hard and the slide would break into two. Please be careful, be gentle. You don't want that to happen at the very end of your gram staining procedure. So just gently press it and make sure you remember the side that you have your cells. You don't want to flip the slide. And I still see some water, so I'm going to put it for a little long time. The goal is to have the slide that's completely dry.